Good morning, everybody. My name is Paul Michael Petersen. I'm a professor at DTU Photonics. It's a great honor for me to welcome you here at DTU campus to participate in this full-day summit on integrative lighting. As you know, light is very important. We cannot see light, but light helps us to see. More importantly, light is a key for life. From cell to complex form of life, from plants to mammals. Being in good health is very important, and it depends on the light. Unhealthy light exposure during the day can result in poor health condition and even in serious illness and cancer or diabetes. We need to quantify the light during the day, the right quality of light at the right time. This is the goal of our summit today. Therefore, I hope that you will help to embrace these challenges for the future in integrative lighting. And therefore, we have today many of the best experts from academia and industry to enlighten the latest development in integrative lighting.
The fact that about 90% of our lives are indoor. Take your own age, multiply by 0.9, and then you find out how many years you spent in your life indoor. Given that fact, and given the fact that that is probably not going to change dramatically in the years to come, of course, we have an extremely important agenda here. How do we create a better life indoor, more healthy buildings, as with respect to light, of course, but also air and sound for that matter. Today we are discussing the light issue, of course.
Right, let's kick off with circadian rhythms. And really, it's sort of stating the obvious, but I think it's worth stating that the 24-hour revolution of the Earth on its axis presents immense complexity to life on Earth. Of course, there's the day-night cycle, which is accompanied by a whole raft of different sorts of ecological changes. Obviously, the amount of light, but also temperature, food availability, and essentially every other factor that determines life on Earth. And in response to this 24-hour revolution, all life on Earth has evolved a response. In humans, of course, we have the sleep consciousness cycle. Sleep is typified by physical inactivity, but as we'll discuss, so many really important things are going on whilst we sleep. And in fact, sleep, the quality of sleep defines the quality of our consciousness. But of course, these are two profoundly different states, sleep and consciousness. And so it's no great surprise that sleep and consciousness are underpinned by an incredibly different sort of biology. But what I wanted to just touch on also is some of the consequences of having a dynamic physiology and knowing about those changes, how they might influence, for example, clinical treatments. Let me just give you a few examples. Here we have some data on the frequency of ischemic stroke. We go back to this image that we saw earlier, and we see systolic blood pressure. And we see that systolic blood pressure has a very sharp rise for around about 6 o'clock in the morning to around about 12 noon. And that 6 a.m. to 12 noon window maps very precisely onto the frequency of stroke. And in fact, between 6 a.m. and 12 noon, there's about a 50% greater chance of having a stroke than any other time of the day. So let's hope we all make it to the end of this lecture. If we do, then we've survived the most dangerous part of the day. Okay, what this timing sort of implies, though, is that when do we take our stroke medication? Many of us will be taking our stroke medication actually 8, 9 o'clock, which is the peak time for stroke. Ideally, we should be taking it, at 5 o'clock before that sharp rise occurs. And so we've got to think of clever ways to deliver medications so that they actually intersect when they're going to be most useful. Also, I guess it's information that could be used to prepare our healthcare services. Knowing that there's going to be that peak in stroke between 6 a.m. and 12 noon, are our ambulance crews and hospitals are suitably prepared to cope with that blip of stroke in time. And really, this leads me to the next point, which is the circadian timing of drug delivery, or chronotherapeutics. This is a long history, and this is a slide that goes back, or data that goes back to 1993. And I deliberately chose an old example because this is knowledge that's been around for a long time. This is a particular study by Rivard and colleagues, and essentially what they did was give chemotherapy either in the morning or the evening. And what was extraordinary is that the risk of relapse was 2.5-fold times higher if the chemotherapy was given in the morning compared to the evening. So same drug, same dose, different time. Those kids that got it in the evening had a 2.5-fold greater chance of survival than those that had it in the morning. This is an old example, but let's now move to a more recent example, and we move from chemotherapy to radiotherapy. And this is the impact of the time of whole brain radiotherapy on overall survival in patients with multiple brain tumors. And it's a randomized controlled trial. And the radiotherapy was given either 8 a.m. to 11 a.m., 11 a.m. to 2 p.m., or 2 p.m. to 5 p.m. And these are the data. So we see that the 8 a.m. to 11 a.m. group all were dead by approximately 6 months. The midday group, 11 a.m. to 2 p.m., all were dead by 24 months, but you see a very clear separation between the morning and the midday group. And what's extraordinary is that the group that had radiotherapy between 2 p.m. and 5 p.m., 10% were still alive at 36 months. So again, same treatment, same condition, different time, profoundly different changes in long-term survival. And finally, an example of time of day of immunization for the influenza vaccine. And this, of course, has high pertinence at the moment with COVID. And so we see here, this was a group of 70-year-old individuals, roughly 70 years of age, and they had their vaccine either in the morning or the afternoon. And we're looking here at the effectiveness of the vaccine in generating an antibody response. The higher the titer, the more effective the vaccination was. And so this is the afternoon group, and we see here's the baseline and here's the follow-up. So, an antibody titer of around 100. But those in the morning had an antibody titer around 400. So again, same vaccine, different time, almost a 4-fold increase in effectiveness by having your vaccination in the morning rather than the afternoon. So the point I'm trying to make is that time of day matters, and the internal clock is dictating so many of our different physiological and behavioral parameters. If we look at some of the changes that we see across the day-night cycle, here we see a number of boxes. The time scale is from 2 p.m. to 2 p.m., so a 24-hour time base. And there's sort of the gray area here is roughly nighttime when we might be asleep. If we just look at one of these hormones and we see melatonin, and we can talk about melatonin in the discussion period. Melatonin, as we will perhaps touch on, is a modulator of sleep. It is not a sleep hormone. But the key point here is that melatonin is rising in anticipation of dark. It peaks, and then in anticipation of dawn, it begins to decline. And what we see in melatonin is replicated in many other physiological parameters. Growth hormone here is almost exclusively released during the first half of the sleep episode. If we don't sleep, actually this growth hormone release is massively truncated. Core body temperature, and in fact this drop in core body temperature might be rather important in sleep initiation. Cortisol, the stress hormone, rises in anticipation of activity. Testosterone is again rising with this dawn morning peak. And indeed, our ability to process information, it changes across the day. It's low point in the early hours of the morning, peaking in the early part of the day, from late morning to mid-afternoon. Okay, well, the point is that these changes, these 24-hour changes, will continue under constant conditions. And they are in large part, but not exclusively, driven or underpinned by the circadian system. And we'll talk about the biological clock and the circadian system in a few moments. Let's now look at the generation of circadian rhythms. So, of course, we see here a cartoon of the brain, and here's the hypothalamus. And if we take a small part of the hypothalamus and we then orientate to the frontal section, we see here where the optic nerves come into the brain, the optic chiasm. Just above that structure is a small paired group of neurons called the suprachiasmatic nuclei. And that's the fundamental heart of the master clock within the brain. The SCN, each SCN comprises about 50,000 individual cells. And what's extraordinary is that each of those cells has its own clock. So here we have the nucleus of the cell, and there's a whole bunch of genes, of course, and those genes, those clock genes, encode proteins. Those proteins are manufactured in the cytoplasm, and then they move into the nucleus and turn off their own genes. So what we have is a clock which is essentially based upon a cycle of protein production and degradation. And that's the fundamental nature of the clock. And we'll look in more detail at the clock towards the end of this presentation. Now, that clock is of absolutely no use unless the internal day is locked on to the external world. So the internal and the external days coincide. And so we see here a light-dark cycle, and we see that that protein oscillation occurs at the same time every day. However, if we were to take away that light-dark cycle, as illustrated here, then the clock will continue to tick, but it slowly becomes misaligned with the real day, with the external world, with the astronomical day. And you see here that the peak is getting later and later and later. Essentially, it's becoming out of sync with the external world. And this illustrates the fundamental importance of light in regulating internal time. But what's important about the light environment? And let me just sort of emphasize where we're going here. I just want to sort of orientate what we're thinking about. So here we see sleep on subsequent days. So this individual was going to sleep and staying asleep for about the same time each day. But when we plunge this person into complete darkness, the sleep cycle, of course, will continue. It's being regulated by the biological clock, but it will drift through time. And we call this the entrained state and this the free running state. And of course, it's light that allows entrainment. What happens if you're not entrained? Well, there's a great deal we could talk about here, but just let me summarize the sorts of disruption that you can have if the clock is not appropriately trained to the light-dark cycle. So it results in very poor sleep. Sleep is misaligned with the external world, and there are short-term effects, there are long-term effects, and indeed there's a third category, that if you have vulnerability to mental illness, you can exacerbate your symptoms. And of course, all these domains, long-term, short-term, mental illness vulnerability, are all linked. I have to say, when I drew this diagram, I hadn't quite appreciated it. It looks a bit like an angel. So I thought I would finish it off and depict the avenging angel of sleep. And what I mean by short-term problems are illustrated here. So microsleeps, 100,000 crashes on the American freeway every year are associated with falling asleep at the wheel. Loss of empathy, frustration, irritability, reduced creativity, poor attention, poor motivation, memory impairment, and negative focus, which we'll talk about later, and irritability. Longer term, the sort of thing that you get in the business community or indeed night shift workers, physiological stress, psychosocial stress, immune suppression. One thing we know about high levels of cortisol is that they can suppress the immune system, which may be the basis for high rates of infection and indeed cancer in long-term night shift workers, for example. Cardiovascular disease, increased stimulant use, increased sedative use to try and compensate for the chronic loss of sleep, metabolic syndrome, and diabetes too. And in mental illness vulnerability, you have mood instability, depression, anxiety, substance abuse, hallucinations, paranoia, mania, and of course, feelings of rejection. So getting the light-dark cycle right to regulate the circadian system, and as we'll see, which in turn regulates sleep, is not just important, it's critically important, because otherwise we experience a range of both short-term and long-term illnesses. But fundamentally, we know that if you don't have eyes, then you can't entrain the clock to the external world. So what is it within the eye that's detecting the light-dark cycle? So here we have a cartoon of the retina, the pigment epithelium, the rods and cones, the visual cells, the inner retina, and these are the ganglion cells whose axons give rise to the optic nerve. And we know that there's a specific tract from the eye to the suprachiasmatic nuclei called the retinohypothalamic tract. And what we showed a few years ago now is that if you have no rods and cones, and we did this first in mice and then in humans, this does not block your ability to regulate the biological clock. So there had to be some other photoreceptor within the eye. We know it's in the eye because if you cover the eyes, the ability to detect the light-dark cycle has gone. And what we and others have shown is that a small number of ganglion cells, which we call photosensitive retinal ganglion cells, are directly light sensitive. They can detect the light-dark cycle, and they are maximally sensitive to blue light because they use a photopigment called melanopsin, which was originally discovered by Ignacio Provencio, who was my PhD student when I was based at the University of Virginia. Okay, so there's a third photoreceptor system within the eye. These are remarkable cells. This is an image showing a bunch of these melanopsin photosensitive retinal ganglion cells, and you see that they form a sort of light-sensitive net across the arc of the eye. We can see them here at high magnification, and they are quite simply beautiful cells. Now, to the point that I think many of you may be most interested, which is how much light do these photosensitive retinal ganglion cells in humans actually need? And it's a complicated story. The critical factors for PRGC light detection and the regulation of a whole range of physiology, including the regulation of the circadian system, will depend upon the intensity of light, the duration of light, and as we kind of will touch on, you can reduce intensity but increase duration, and so these two interact. The wavelength or the color of light, the time of day, whether it's dawn versus dusk, this is really important and we'll touch on that. The task. We've talked about the circadian system, but those photosensitive retinal ganglion cells are not just regulating the clock, but a whole range of other sorts of pathways, such as alertness, such as the regulation of melatonin, and we'll touch on that. So and the thresholds vary between these different tasks. The light history, this is really important, whether you spend most of your time outside versus inside. That can change the gating of your sensitivity to the light-dark cycle. And indeed age. There's good evidence suggesting that young people are more sensitive to light in terms of the regulation of their clock versus older people who are less sensitive. Let's kick off with intensity. So we, as you will be more than aware, are exposed to a very broad range of light exposure within the environment, going from approximately 0.01 lux, which would be something like moonlight, going through to a candle at 1 meter, which would be one lux, museum display case and sunset about 50 lux. Office lighting varies enormously, but in most instances it's between sort of 300 to 400 lux if you're lucky. Near a window, perhaps 3,000 lux. Shade outside, 10,000 lux, and even in England you can get up to 10,000 lux on a full sunny day. Now the rods provide us with our dim light vision. The cones give us, of course, our color vision and can operate over this range here. So under normal circumstances, there's plenty of light in the office for good color vision. Those photosensitive retinal ganglion cells need more light. Now, again, it's complicated because it's not really fair to compare the rods and the cones with the PRGCs, because here you have fractions of a second of light exposure. Here you need not only bright light in the 100 to 1,000 lux range, but also of a long duration. And so, for example, light boxes for the maximum effectiveness are in the sort of 5 to 10,000 lux range with an exposure of 30 minutes. The point I'm trying to make is that the PRGCs relative to the visual cells are incredibly insensitive to light, but we'll come back to the visual cells fairly shortly. So most of the time we're exposed to sort of, let's say, 300 to 400 lux in the office, fine for color vision, but it's not often a robust enough signal to give robust entrainment for the circadian system. Let's now think about wavelength. Well, we used to think that the photosensitive retinal ganglion cells just have no interaction with the rods and cones. And we now know that that is not the case. So the rods and cones can communicate with the photosensitive retinal ganglion cells indirectly via the other neurons in the retina. So the bipolar cells from the cones have a direct connection with the PRGCs. The rods have a rod bipolar, which then talks to an amacrine cell, which then talks to a cone bipolar. And so that's how the rods can influence the PRGCs. So under normal circumstances, we have to factor in a modulation by the rods and cones. Problem is, we don't fully understand how those interactions occur. So the point I'm trying to make is that here is the maximum sensitivity of the PRGCs, but there's potentially an input from the rods, from the short wavelength cone that we have, peaking around about 420 nanometers, one of the longer wavelength cones peaking around about 530, and the longest wavelength cone that we have peaking about 560. So the point I'm trying to make is that there's potentially a broad spectral input to the PRGCs and their modulation of the circadian and other systems. And it's also not necessarily additive. There's some studies from primates which suggest that the short wavelength 420 cone might actually be inhibitory. And at the moment, we simply don't know, but we've got to be sensitive to the fact that it's more than just blue light peaking at 480 nanometers that's regulating the clock, for example. If we now look at the time of day, and I think this is a really important point because this is often missed in models of trying to understand how light interacts with the human circadian system. When you see light really matters. And let me illustrate that. Here we have a day-night cycle, and this is when the individual's asleep on subsequent days. Now, if we see light at dusk, this delays the clock. It makes us go to sleep later the next day. But when we see light at dawn, it advances the clock. It makes us go to sleep and get up earlier the next and subsequent days. So under normal circumstances, when we were all agricultural workers, we got a symmetrical exposure to dusk light and dawn light, and we stayed more or less aligned to the light-dark cycle. But many of us now are exposed to only one part of the light-dark cycle. And let me show you a paper we published a few years ago now on university students. And to summarize those data, we showed that can everybody hear me okay? Are you all there? Can you hear? Okay, I can't hear you. I'm assuming you can hear me, yeah?
We can hear you clearly.
Oh, fantastic! Jolly good. Okay, great. So this is a study which we undertook on university students, and we see that they are sleeping through dawn, so they don't get their dawn exposure, which would make them get up earlier. But of course, they're awake and they get dusk light exposure. So the net effect is to go to sleep later. So part of the lateness that you see in young people is that they're missing morning light exposure, dawn light exposure, but getting dusk light exposure, which is pushing the clock to a later time. Again, the point I want to make is that getting light exposure at the correct time is critical for regulation of the clock. And this feeds into something I'm going to touch on later, but I wanted to deal with now, which is a really important component of the way that light regulates the clock, and it's called the phase response curve. And let me try and illustrate what I mean. Here we have a light-dark cycle, and this is for, let's say, a human subject, and here we see subsequent days of activity. If we plunge this individual into complete darkness, then, as we discussed before, the clock free runs and it drifts through time. Now, if we give a pulse of light in complete darkness, when the clock thinks it's the middle of the day, the middle of the activity period, that has no effect in shifting the clock. And this is expressed in the following way, in the phase response curve. This is getting up early, if you like. This is getting up later, and we see here there's a dead zone. So when the clock thinks it's the middle of the day, we're pretty unresponsive to light. But if we see light at dusk here, then you see it causes a delay. And the further we go into the night, we get a bigger delay. Whereas in the morning, if we see light at dawn, we get an advance, and that's plotted in this way. So this is the phase response curve. The exact shape of the phase response curve in humans is still being argued about. There are some who say that there is no dead zone in the human phase response curve, and it's essentially a straight line through here. Others argue that humans, like all other animals, actually have a dead zone. The point I'm trying to make is that you must take time of day and this phase response curve into account into looking at the effects of the clock, light on the clock. The task, I touched on this. We now know that those photosensitive retinal ganglion cells are plugged into a whole range of structures within the hypothalamus and the thalamus. So we know that they're regulating, of course, the circadian system via the SCN, the VELPO, which is involved in sleep regulation, the lateral hypothalamus, which is sleep, alertness, metabolic regulation, and reward systems, the subparaventricular zone, which is integrating circadian and metabolic information. There's the lateral habenula via the ventral tegmental area, regulating dopamine release, via the raphe serotonin. There's a projection to the lateral geniculate nuclei, and that, of course, is potentially mediating vision effects, as is a projection to the superior colliculus. So there's a huge role for these PRGCs in regulating biological responses to light. And, of course, via the sympathetic nervous system, the light-dark cycle can either directly or via the circadian system regulate sympathetic control. Okay, what I wanted to really emphasize here is that we must take intensity, duration, wavelength, time of day, task, light history, and age. And really, we need good studies. We need to understand the ecology of human light exposure. And with this information, we can begin to develop models of trying to understand how environmental light interacts with our biology. And I know that's been a key topic of the meeting. The sleep state, the quality of the sleep that we get defines effectively how we're able to function during the consciousness and the wake state. I just want to illustrate the importance of sleep in two domains. One is in information processing. This is a beautiful set of experiments from Jan Born's group in Germany. And he developed a problem-solving task. And he introduced it to one group in the morning, and they performed the test that afternoon. And you see that about 20% of the group were successful in solving the problem. The next group were introduced to the task in the morning, but they then performed it the following afternoon, but they were deprived of sleep, so they had no sleep. And you see that around about 20% of the group were able to solve the problem. So he'd nicely controlled for fatigue here. The third experimental group was that they were introduced to the task in the morning. They then performed it the following afternoon, but they were allowed to sleep normally, and 60 to 70% of the individuals were able to solve the problem. So this really illustrates the point that sleep promotes the ability to come up with novel solutions to complex problems. And the reason I'm talking about this is because, of course, there's an intimate association between circadian system and light and the regulation of sleep, as we'll see later on. My final example is the interaction of sleep, memory, and emotion. Now, this was from Sickhold's group, a very nice paper, where he compared two groups. One group that had slept normally and another group that had not slept for one night. So when they performed the tests, they had been asleep for they had been awake for 36 hours. They had been sleep deprived for 36 hours. And what they were asked to do was to remember words with a different emotional content. So positive words, love, happiness, joy; negative words, hate, war, crime; and neutral words, things like cotton or shirt or something like that. Let's look at all the words and their retention in the two groups. And perhaps no great surprise, the sleep-deprived group showed a diminished ability to remember words in total. But let's now divide that into the neutral words, the negative words, and the positive words. There was a trend to remember fewer neutral words, but it wasn't significant. There was a trend to remember fewer negative words, but it wasn't significant. The overwhelming result was that tired brains failed to remember the positive. And this sort of illustrates the point very nicely that the tired brain has what's called a negative salience.
Okay. Hi, Russell. Thank you so much for your talk.
Oh, hello. Is that Joachim? Hi.
How are you? Thank you so much, Russell.
Great pleasure.
My question is extremely fascinating with the whole wake episode.
Yeah.
I'm wondering whether at the moment
I'm sorry to jump in. I mean, really, the key point there is, we're building these models about how light is interacting with the circadian system. And clearly, we need to factor in duration of wake, because the longer we've been awake, the less effective that light will be.
But my question then becomes, that's fascinating. So light history is important here.
Yeah, absolutely.
The next thing is, have you looked into how, let's say, non-photopic sightkeepers then interact in that whole wake state? So here I'm thinking about food timing. I'm thinking about activity, so in muscles and so on. Whether that then has an advanced input to that wake state.
Yeah, no, I think that's a really important point. And whether the food timing, because just sort of a little bit of background, we do know that eating at the same time every day can influence the timing of the circadian system. And whether that is via a similar pathway that is activated by adenosine, I do not know. And I think it's an incredibly interesting question, because we've talked about, sleep-wake history, and we've talked about light, but we perhaps should now think about the way that feeding might interact with those molecular pathways. I think it's a really good point.
Okay, so thank you so much for doing this, Russell. It was,
great pleasure.
So the eye has two functions, just like the ear, where the ear gives us the ability to hear and keep balance. So does the eye have two functions. It makes us see and see pictures and our surroundings, but it also tells us some very important cues about time. And this is something that we need to take into consideration when talking about light going forward. And we need to integrate it into how we make good lighting. So one of the things that basically has happened is that CIE has introduced the term of integrative lighting to try and deal with this. But why now? That's a good question. I would argue, and this is what I'd like to talk about in this presentation, that there are at least 6 key drivers to this. One is the need for solving a problem. The lighting science, which tells us, and the science which tells us we need to take this into account. Then there's innovation in measurements, which I'd like to get into. There's lighting technology. There's early frameworks and recommendations, and these are happening as we speak. And then there are early examples and use cases that we can learn about. And this is what I'd like to talk to you about today. So first of all, the need for solving a problem. Light and circadian rhythms are hardwired into plants, animals, most species on Earth, including humans. And for millions of years, the natural light-dark cycle and the changes in light-dark cycle, which is very scheduled, our body has basically adapted to this. But things are happening and things are changing. New modern lighting systems, the illumination of light at night, and the fact that we spend a lot of time in buildings away from, for example, daylight, might actually interfere with these very, very ancient systems. And this might mess up our biological clocks, our circadian system, and a lot of other physiological responses, to a point where American Medical Association in 2016 actually adopted political guidelines to the reduction of reducement of harm from high intensity streetlights. Another quite interesting fact is how modern lighting can be summarized. This is from Thiel Ronnenberg's group in Germany, showing that over a 10-year period, we basically get less light, less daylight, we sleep less, and then we get social jet lag, being the difference between mid-sleep on weekdays and weekends. So our circadian clocks are not synchronized. So there's a problem here that we need to solve, and light is a major benefactor of this. So what about the lighting science? What does that tell us? Around 20 years ago, we learned of a new receptor system within the eye, an EPRTC or PRTC system with the photopigment melanopsin. The conclusion of that is that there's a new revolution within light and lighting. It's not only a visual aspect, we also have these non-visual aspects driven by intrinsically photosensitive retinal ganglion cells. What's important to mention here is that we actually do see quite a good correlation between these EPRTC cells, although there are many other involved in circadian entrainment, circadian phase shifting, and melatonin suppression. And we see, this is also where we see the lighting community targeting. But what I want to stress very carefully is that we know from research now that these EPRTC cells, they stimulate a host of neuroendocrine and neurobiological responses to light. And we need to take this into consideration when we do lighting. And this is our subjective alertness. It's cortisol stimulation. It's our pupil light reflex. It is photo entrainment and seasonality, and even food anticipation, mood, and hippocampal learning. So it's light is quite important for us. So to mitigate some of this, basically from within lighting industry, we've seen a host of concepts to target this with a new guideline in their vocabulary, which is integrative lighting. And again, this is adopted in the CIE vocabulary now. Now, we also hear about concepts such as human-centric lighting. And just to give you an example, this was published, I think it was actually last week, by Hauser and Boyce and Saiter. And they basically looked into myth, magic, or metaphors of integrative lighting and human-centric lighting, concluding the following. The phrase integrative lighting avoids the hype of human-centric lighting and encapsulates what lighting aspires to be. Something old, which is core ingredient of good lighting, driven by good design teams, lighting designers, etc. And then the fact that light is still for vision, to be able to see. Light is still for visual comfort, and it is as important as ever. But there's a new thing that we need to integrate, being the new, which is an awareness of non-visual responses to light, neuroendocrine and neurobiological responses that are important for human health. And then the realization that to meet this, we need to make design solutions within lighting to contrast day and night. And I would argue personally that this is one of the best examples of how a schematic framework could look like. This is actually from Karin Smulders from 2014, so it's nothing new. And what she basically stipulates is that we can use the image forming light and the non-image forming light. And the image forming light being good visual performance, visual acuity. Visual experience, this is where you hire a lighting designer. Then the circadian effects, as we saw in melatonin, phase shifting. And then acute circadian effects. This could be light's influence on alertness. But we need to be able to articulate what it is we want out of these lighting systems to be able to target this. Further, CIE has issued a roadmap for helpful interior lighting. And what's interesting about this is that they also describe classified influencers of these processes. What about the individual, the age of the individual, visual impairment, stressors and other factors? What about amplitude of daily patterns? When we talk about circadian lighting, do we sufficiently talk about seasonality? I would argue not. And we need to be much better at that. And then also including these acute non-circadian effects in neurophysiology. And then also the changes within the retina. So what about these non-visual effects? Are they proven? Do we know enough? There's a lot we still don't know about this system. It's really complex. However, we are beginning to arrive at evidence that light is relevant for human behaviors, health and well-being. And we do see meta-analysis being published. This is from 2016, showing a great correlation with acute alertness, a great correlation with melatonin suppression, and a great correlation between concentration and performance using the 2D test, including depression scores. So the use of non-visual light is something that we should focus on. So what about innovation in measurements? How do we measure all of that? How do we check? How do we talk about it? Well, before 2002, we looked at measurements in the visual age. So we know all of these: Kelvin, lux, illuminances, glare, etc. These are as important as ever, and we need to focus on them. But I would argue that we've seen sort of two directions within how we measure these non-image forming approaches to light. This is a rough schematic, but around 2002, when we first encountered these EPITC cells and melanopsin, around 2011, we saw NC describing this melanopic response. Bale and Lucas improved on this for the melanopic response. And then in 2014, we saw a breakthrough study from Lucas et al. Advocating that because of the complexity of photoreception within the retina, we need to calculate and measure light on each of the 5 photoreceptor systems within the eye. This is something that EU and CIE has adopted in a number of standards, and the newest CIE 2018-026 is actually adopting this, telling us that we now need to measure light on each of the 5 photoreceptors within the eye. This also means that we have the visual age and now the non-visual age. So couldn't we just use photopic locks to anticipate these non-photopic and non-visual responses? The answer is no. This is basically melatonin suppression. And as you can see, the melanopic locks is much better at prescribing melatonin suppression than the photopic locks value. So we need to measure light in a new way. And I would again argue we can also see that cyanopic and rhodopic is basically stimulating melatonin suppression as well. So please start to use the new toolbox. This is also what CIE is describing, recommending proper light at the proper time, concluding that two important well-studied human non-visual responses are out there, melatonin suppression and pupil control. And the evidence is accumulating that overall light sensitivity of these responses are largely controlled by melanopsin-based photoreception. If you want a high stimulation, you go for a high melanopic EDI during the day for alertness and circadian rhythm and good sleep. And in the evening, a low melanopic EDI to consolidate sleep. And to be able to use this, CIE has published this CIE S026 2018 methodology of optical radiation of EPRTC influenced responses to light, which basically measures all 5 photoreceptive fields within the eye, L, M, and S cones, rods, and a melanopic response. And together with this standard, CIE has published a toolbox, which is basically accessible online, so everybody can get access to it, which basically can take light spectrum and CIE approved light sources and give you basic quantities in irradiance, illuminance, and log photons equivalent of all 5 photoreceptive fields. So please start using this. So why should we use this? Well, this might be one of the reasons why. Depending on what we are measuring, depending on what light should do, whether it's pupil light reflexes, whether it's visual acuity, whether it's concentration, phase shifting of circadian clocks, or melatonin suppression, different alpha-opic EDIs or different photoreceptors are involved. And we need to be better at articulating this when we prescribe lighting. So can we use this? Well, this is a study done by Brown from University of Manchester. It came out in 2020. And they basically looked at 17 of the classic studies within circadian physiology and basically found that melanopic EDI or melanopic illuminance was the best predictor for circadian impact on environmental illumination. And that this supported a very strong use of these in the guidelines and regulating light exposure to benefit human health in future lighting design. There's a very, very nice correlation in these studies. So we should start using this toolbox to articulate the non-visual effects of light. Measurement. What about the lighting technology? Well, we see changes in lighting technology as well. We see dynamic lighting systems. These are used today. These are called dynamic lighting, døgnrytmelys, circadian lighting, etc. But we also see changes in, for example, LED lighting technology. This is basically a study by Joachim's group in Basel, who has looked at these daylight LEDs, which are new LEDs that have a broader spectrum. And what you can see is, compared to conventional LEDs, these daylight LEDs don't have this dip in the middle of the blue spectrum, which means that it has a 21% stronger melanopic strength than conventional LEDs. And this study done by Joachim's group has actually showed that this could give better visual acuity, higher delta EEG under deep sleep, and better wakefulness during the day. These same LEDs have been used in this study, where they have looked at the dynamic of light. So much light during the day, low light during the evening, and then compared to stationary lighting. And what they actually found was that one of these dynamic LED lights, less melatonin suppression was found 1.5 hours prior to sleep, less vigilance, so more sleepiness, and shorter sleep latency going to sleep earlier. So the dynamic of the light is important, and perhaps even the lighting itself. So what about early recommendations and frameworks? How much light is needed when we look at light at night. First of all, light at the eye matters. The relationship between melatonin and alertness at night is quite well established. And again, this also means that we can measure on some of these photoreceptor fields within the eye and get some good insight into, for example, melatonin suppression. Melanopic lux is best correlated as a single receptor. And again, we also see that under controlled lighting conditions, it's actually not that much light needed to actually suppress melatonin. Then the question becomes, what about an optimal daily light exposure pattern, a daily light diet? And what about the acute effects of light? Some studies have indicated around 1,000 lux. And actually last week, this was published, which is basically from Yvonne Decourt's group in Eindhoven, showing a literary review on dynamic lighting of 14 studies that have used dynamic lighting, or what we call circadian lighting systems. And they describe this as being color, temperature, and illuminance varying during the day to a present protocol, aiming to support or even enhance natural rhythms and employ alertness. What was interesting about this report or this review was that what they basically found was that the effectiveness of dawn simulation on sleep inertia and sleepiness has been well established, and they could see in these studies an increase in morning light exposure may actually support the circadian system. The other thing they found was a horizontal illumination of more than 1,500 lux at the end of the morning seems to be related to a shorter sleep onset compared to 1,000 lux or under. And then again, what they concluded was that there's so big a diversity in protocols that you don't get insight to. So it's really, really hard to compare across studies. So we need to be better at describing what we want. And this is also a broader consensus we see from scientists, who has basically submitted 7 important factors that we need to take into account when and how we report these non-visual effects. And this is out there and it can be done and it should be integrated, I would argue. And this is basically also using the alpha-opic EDI as an offset. Further, there are initial frameworks. This has not been published yet, but it will soon in a book. But we're starting to see proposed frameworks for lighting episodes throughout the day, where different sort of light can have an influence on the circadian system. And basically what we want to do with this is to allow for a more informed discussion and a more detailed discussion instead of just lux and Kelvin, but what is it you want to achieve with your lighting system? This is together with Katarina Wolf, and it will be published, I think, hopefully later this year. So what about the integrative lighting and the individual and complexity of the environment? How do we take that into account? Lighting is hugely complex and how we encounter it. First of all, we need to take into account huge individual differences. Just look at age. You know, this is a 32-year-old compared with a 72-year-old, and you can see huge responses here. And the other thing is that there's an inter-individual sensitivity within humans. Do we take this into account when we adopt circadian lighting? Further, what about the visual system when we actually do circadian lighting or when we do non-visual lighting? This is actually, I think George Brainerd did some studies on astronauts a couple of years ago, but otherwise I haven't seen this yet. But this is a study just published, and they looked into this blue depleted light at night into circadian system and promoting sleep within a hospital environment. All good and well. But what they also did was they used the Munsell Q 100 to look at the amount of errors in these lighting settings. And you could see that in this blue depleted light, when you lose spectrum, you also make more color mistake. So we need to be aware that non-visual lighting system and visual lighting systems are combined. Next, what you see is not what you get. This is a study by Smolders and Decourt, who basically looked at a lighting system where they introduced a sort of dynamic lighting system where they increased the horizontal lighting level to 900 lux, giving 300 lux vertically at the eye. And then they looked at individuals and their light exposure in this room. And what they found was that some of these had encountered this increased light exposure, but many of them showed virtually no signs of the additional lighting. So our light history and how we move around in spaces we need to take into account. And again, this has also been seen by Russell Foster's group and Pearson and Anderson, who a little less than a year ago did a study on the spectral diet of humans and actually advocated that we need to take light history for the circadian system into account, the non-visual system, measuring light over time to be able to adopt well exposure patterns. And the reason for that is that patterns we are exposed to of light are inherently messy. We can't assume that we can install a human-centric lighting system and then that people will be there all the time. So we need to get insight into the light history, simply put, if we want to be precise. And we actually see some initiatives in this. Daniel talked about Ocutune and the product we're developing in collaboration with Paul Michael's group, because we actually think it's quite important to measure this light history and to be able to adapt to it. But we also see other companies and other research institutions looking this way. And when implementing this, there's a something we need to look into, which is basically the accuracy and the acceptance from users, and we're also taking that into consideration. So what about the early examples of use cases? This is an example. You see here, let's look at a scenario where you have blackout curtains, you have a light source of warm LEDs, giving you 500 lux at the eye, or at the reading environments, and measurement of 6 lux at the eye. Would that potentially have an influence on the circadian system? Many things are involved, but this is a rough schematic of how it could be done. So when we look at the circadian system at 11:30 or 10:30 at night, for this individual, we would be within the subjective night, and hence we should not disturb melatonin. So by using the new toolbox, we can actually do a calculation of all 5 photoreceptors within the eye because we have the light spectrum from the light source. And here we can see the alpha-opic responses of, this is basically using Lucas's toolbox, but of a 6 lux photopic lux light source. And then using the knowledge about the the melatonin suppression, we can actually see that this would sort of be okay to not suppress melatonin. So we can begin to make these calculations. So to conclude, light has a visual and non-visual effect, which can't be ignored. We need to take this into account, simply put. Agreement was reached in 2018 on how we measure these things in an EU way. And this is basically to focus on alpha-opic light responses for all 5 photoreceptors fields within the eye. Using melanopic daylight illuminance or illuminances is a good predictor for at least melatonin suppression, melatonin circadian phase shifting, and also the alerting responses to light. Measuring 5 alpha-opic EDI and recording light over time to get light history into the equation is vital. And we are seeing some initial products on its way here, and that needs to be integrated. Guidelines and standards and reporting guidelines are emerging. So I would argue use them and use the toolbox when doing lighting. And then when talking about circadian lighting, døgnrytmelys and HCL in a Danish context, we simply need to get a more common language of what we're talking about. Otherwise, we don't know what we are prescribing within light. And this is also why there's a book on the way to take up that discussion. So that will be published later this year. So thank you for your attention. And any questions?
No, the question is just when you say it's very important to measure light history, and I fully agree on that. But how long should that history normally be? Be 1 day, a week, or yeah?
If you start with 1 day, that's better than most. Yeah. So start there. But as we know that melatonin, for example, just the it's a dark hormone, then that's also a seasonal sidegaper. So. And I would argue we actually see some studies done within lighting communities showing that if you implement a static or a dynamic 24-hour lighting system, it basically has the opportunity to override seasonal sight gapers. And I would argue this is wrong. So basically talking about circadian lighting as being one 24-hour period is totally wrong. And we need to take that into consideration when we do circadian lighting.
My question is just if there's a lot of confusion about the language, and I know that, but do you have a good Danish word for integrative lighting?
The short answer would be no. I would put it this way. I understand the need for trying to put all of this into a box. And I also argue to some extent we need to do that because otherwise it becomes too complex to articulate. But I think sometimes it becomes too simple in what you want to achieve. And when you do that, you simply miss what it is you want to achieve. And I think sometimes you do that when you just call it human-centered lighting. Again, there's two things I think you can do. I would argue if you do a circadian lighting system or an integrative lighting system, if you hear someone only talking about lux and Kelvin, I would be a bit wary. And then I would say, what about the non-visual effects and how do you actually get insight into this? That's one thing. So we can start to begin to differentiate between the visual responses, being lux, Kelvin, all of the UGR, all of the things we know, the quality and the visual effects. And then we have the non-visual effects. If you just take the pupil light reflex, for example, 3 photoreceptors are involved. So just by using lux, you won't be able to articulate anything about the pupil light reflex in detail. You need the alpha upping EDI to be able to articulate that. Yeah. So it's new times within lighting.
Yes, and what I'm just looking for is also specifying lighting, that you have both aspects.
I agree. I would argue, this is me personally, I would argue that the best schematic, as I also showed you here, is Kind Smolder's description and schematics from 2014. I still think this holds water. And I think it allows you to at least try and differentiate between these image forming and non-image forming effects. For example, what about light during the day when the circadian system and melatonin, for example, is very low? Then you can't just focus on melatonin. So you need to be able to differentiate between these acute non-circadian effects and the circadian system.